of respiratory gases, oxygen and carbon dioxide, within the blood. So oxygen transport is carried in two ways. It's about 1.5% of it is dissolved in the plasma, and 98.5% of it is loosely bound to iron atoms in the hemoglobin molecules of red blood cells. So the association of oxygen and hemoglobin is a little bit complicated. It's a cooperative association when you're binding, and it's also cooperative when you're unloading, and it's important for us. But each hemoglobin molecule is composed of 4 polypeptide chains, and each one of those chains has an iron-containing heme group, which means that a hemoglobin can transport 4 oxygen molecules. If it is got oxygen on it, we call it oxyhemoglobin. And if it does not have oxygen on it, we call it either reduced hemoglobin or deoxyhemoglobin. So in the lungs, oxygen binds to hemoglobin and hydrogen ions are released. And in the tissues, that oxygen is released from the hemoglobin. So loading and unloading is facilitated by a change in the shape of hemoglobin. The shape of hemoglobin is important, and that's why as one oxygen binds, it begins to change the shape of the hemoglobins, and that increases the affinity for oxygen to the other 3 hemoglobins. The second one helps with the last 2, and the reverse happens when one is released. The affinity for oxygen, the other oxygens, decreases. So if it is fully saturated, it has all 4 oxygens carrying, all 4 heme groups carrying an oxygen, we call it fully saturated. And if it has less than 4, we call it partially saturated. There's only 1 or 2 or 3 oxygens. And that rate of loading and unloading is going to have to be very tightly regulated. That helps ensure adequate oxygen delivery to the cells. And the things that are going to influence hemoglobin saturation are the partial pressure of oxygen and other factors such as temperature, blood pH, partial pressure of carbon dioxide, and the concentration of a molecule called BPG. Oxygen partial pressures have a very heavy influence on binding and releasing of hemoglobin. So the percent saturation of hemoglobin can be plotted against the oxygen concentrations. Because of that cooperativity of binding and unbinding, you do not get a linear curve as you might expect, but it is a S-shaped curve, a gentle S shape. We'll see some later on. And that is referred to as the oxygen hemoglobin dissociation curve. So in the arterial blood, the partial pressure of oxygen is about 100 millimeters of mercury, which means it contains about 20 milliliters of oxygen per 100 milliliters of blood. In that case, hemoglobin is 98% saturated. And if you increase the oxygen levels even more, like when you are deep breathing, you're going to get a minimal increase in O2 binding. The binding is already nearly complete in arterial blood. In the venous blood, the partial pressure of oxygen is 40 millimeters of mercury, and it has only about 15% of the volume that it would have in arterial blood. But that doesn't really do a lot to the hemoglobin. The hemoglobin is still 75% saturated, and this is what we call the venous reserve, the oxygen that's in venous blood. That oxygen can still be used. This is one of the reasons that CPR is now being taught, and they're not worrying so much about breaths, because there's plenty of oxygen in the venous blood until we can get patients where they need to be. Other factors would include increasing temperature, increasing hydrogen ion concentrations, which would mean becoming more acidic, increasing levels of carbon dioxide and BPG. All of these can result in a decrease in hemoglobin's affinity for oxygen. These are the kinds of conditions we would find in the systemic capillaries, and that helps to enhance the unloading of oxygen, causing that shift in hemoglobin dissociation curve to the right. If we decrease those factors, we're going to decrease oxygen unloading from the blood. And here is that what's described as an S-shaped curve. So here is a look at the binding of oxygen at normal body temperature, about 38 degrees Celsius. If you go this way, you shift to the right. You go this way, you shift to the left. Here we see the same thing with carbon dioxide. BPG is produced by red blood cells during the process of glycolysis, and BPG levels are going to rise when oxygen levels are low. So as cells metabolize glucose, they're using O2, and that causes an increase in partial pressures of carbon dioxide, an increase in hydrogen ions, so a declining pH, and that will help weaken the bond between oxygen and hemoglobin, and that is referred to as the Bohr effect. And that helps ensure that oxygen unloading occurs where it's needed the most. Heat production in tissues that are actively metabolizing increases or indirectly and directly decreases the affinity of hemoglobin for oxygen, which again helps to ensure that oxygen gets unloaded into tissues that are actively metabolizing. Carbon dioxide, on the other hand, is transported in 3 different ways in the blood, including dissolved in the plasma, bound to hemoglobin, and in the plasma but in a different form. So about 7 to 10% of the CO2 is dissolved as CO2 in the plasma. About 20% is chemically bound to hemoglobin. It's not the same binding that you would have for oxygen. It's not bound to the iron. It binds to a globin part of hemoglobin, and this is referred to as carbaminohemoglobin. It is a reversible reaction, so CO2 plus hemoglobin, this should be an arrow, would give you carbaminohemoglobin. But most of the CO2 is dissolved in the plasma as a bicarbonate ion. So about 70% of the CO2 is actually bicarbonate, HCO3 minus. That happens because carbon dioxide will combine with water to give you a chemical called carbonic acid. Carbonic acid will quickly break down into a hydrogen ion, which is why this is an acid. It donates a hydrogen ion to solution, and bicarbonate ion. So this HCO3 really represents the CO2. And we're going to use that to regulate pH within the blood, as we'll talk later on. That reaction occurs primarily in red blood cells. There's an enzyme called carbonic anhydrase that reversibly and rapidly catalyzes that reaction. The combination of carbon dioxide and water to give you carbonic acid. In the systemic capillaries, after the bicarbonate is created, it diffuses from the red blood cells into the plasma. That outrush of bicarbonate from the red blood cells is going to be balanced because to make up for that negative leaving, a negatively charged chlorine ion will move into the red blood cell, and this is called the chloride shift. So we have oxygen bound to hemoglobin being released to go to the tissues. Carbon dioxide is being generated in the tissues. It's getting into the red blood cell. It is combining some with hemoglobin to give you carbaminohemoglobin. Some of it is going to combine with water using the enzyme carbonic anhydrase, which would give you carbonic acid, which would break down into bicarbonate and hydrogen ions. As that bicarbonate leaves, chlorine will go in. That's our chloride shift. And then there is also some CO2 dissolved in the plasma. And this chemical reaction, the combination of carbon dioxide and water to give you carbonic acid, can happen without carbonic anhydrase, without the enzyme, but it's a slower process. And these will be all found in the blood. Pulmonary capillaries, the process is going to happen in reverse. Bicarbonate is going to move into the red blood cells. Chlorine is going to move out. chloride is going to move out of the red blood cells back into the plasma. In that case, the bicarbonate ion will recombine with the hydrogen ion to form carbonic acid. That carbonic acid, H2CO3, is then going to be split by another enzyme called carbonic anhydrase back into carbon dioxide and water. The carbon dioxide would then diffuse into the alveoli. So in the lungs, carbon dioxide is leaving, and oxygen is getting reloaded. The Haldane effect has to do with the amount of carbon dioxide being transported is affected by the amount of oxygen, the partial pressure of oxygen. The lower the partial pressure of oxygen in hemoglobin oxygen saturation, the more CO2 can be carried in the blood. That's going to reduce hemoglobin. That means that reduced hemoglobin buffers the hydrogen ions, and it forms carb amino hemoglobin more easily. And this process encourages carbon dioxide exchange at the tissues and at the lungs. So at the tissues, more carbon dioxide enters the blood, more oxygen dissociates from hemoglobin. That's the Bohr effect. And as hemoglobin releases oxygen, it can more readily bond with CO2 to form carb amino hemoglobin. The influence of carbon dioxide on blood pH. There is this carbonic acid bicarbonate buffer system that helps blood resist changes in pH. If hydrogen concentrations in the blood go up, so the blood becomes more basic, the excess hydrogens can be removed by combining it with bicarbonate to form carbonic acid, which can then go back to carbon dioxide and water. On the other hand, if hydrogen ion concentrations begin to drop, it becomes more acidic, the bicarbonate dissociates, releasing hydrogen ions back in. And I got those backwards. Let's go over that one more time. So if hydrogen ion concentration in the blood rises, you have an excess of hydrogen ions that is making it more acidic. So the bicarbonate would reform carbonic acid, and that would go back into CO2 and water. On the other hand, if hydrogen ion concentrations begin to drop, this would be more basic. The carbonic acid would dissociate and release hydrogen ions to bring it back to where it should be as far as pH goes. So therefore, that bicarbonate is considered the alkaline reserve of the carbonic acid bicarbonate buffer system. Changes in the respiratory rate can also affect blood pH. So slow, shallow breathing causes an increase in carbon dioxide in the blood, which would lead to a drop in blood pH. Rapid deep breathing causes a decrease in carbon dioxide in the blood, and that would cause a rise in the pH, be more basic. So changes in ventilation can be used to help adjust pH when the disturbances are caused by metabolic factors. So breathing is going to play a major role in the acid-base balance of the body. Hypoxia is when you have inadequate oxygen delivery to the tissues, and that can result in cyanosis. These hypoxias can be classified based on their cause. You could have anemic hypoxia, which would mean there are too few red blood cells, or the red blood cells you have are abnormal, or do not contain enough hemoglobin. You can have ischemic hypoxia, which is impaired or blocked blood circulation. Histotoxic hypoxia, and the cells are unable to use the oxygen, as in the case of metabolic poisons such as cyanide. Hypoxemic hypoxia is an abnormal ventilation, a pulmonary disease, and you get low levels of oxygen in the air. Carbon monoxide poisoning is because hemoglobin has a 200 times greater affinity for carbon monoxide, the chemical carbon monoxide, than it does for O2. Patients who are suffering from carbon monoxide poisoning will get headaches, become flushed, and they have some very distinguishing coloring characteristics, rosy coloring in their lips. And the only way to treat carbon monoxide poisoning is to use a hyperbaric chamber and try to force the carbon monoxide off the iron so that oxygen can be rebound to it. And that will take us to control of respiration, which I will